So thank you for the introduction. I have nothing to disclose. So I'm going to be talking about high temporal resolution fMRI. And so this is just an image here of data that we acquired at relatively high temporal resolutions. So classically, fMRI would acquire with TRs of maybe 2 or 3 seconds. And it's now become possible to do this with TRs of just a few hundred milliseconds. And when this happens, I guess the video is not playing here, you can see that actually the dynamics of fast fMRI data are quite complex. You can see pulsing in the ventricles, you can see flow in vessels. And so there's some qualitative differences as you speed up your acquisition in terms of what's present in your data and how you should be analyzing it. And so the development of these techniques has been, in many ways, very exciting, in part because what we actually typically want to study with fMRI usually is relatively fast. So oscillations, for example, in neural activity are usually in ranges of above 0.1 hertz, so on timescales that we're really starting to move into with these faster acquisition techniques. And then many of the users of fMRI for cognitive neuroscience are studying things like, for example, perceiving a sentence or deciding what to attend to in the world that really unfold on these timescales of hundreds of milliseconds. And so this speaks to the potential of faster imaging to really bring fMRI to ask new kinds of questions in neuroscience. Even if what you're studying isn't one of these fast signals and is relatively slow, there's also this very nice property of fast fMRI that you simply acquire more data. So here's an example of a visual task we ran where we had a stimulus coming on and off. And you could see that if we only had data every couple of seconds, it might be a little bit difficult to tell whether activity in this region is actually related to the stimulus that we presented. But if we sampled 10 times faster with a TR now of 227 milliseconds, it becomes very clear just from looking at your data alone that there's actually a really nice time-locked signal in this region of interest to the stimulus that we were representing, even though the stimulus itself is slow. And so these are often the two major advantages of fast fMRI. One is that the neural activity we'd like to study is actually usually fast. But there's, of course, this major caveat, which is that what we measure in fMRI is not the neural activity, we measure the hemodynamic response. And so to actually understand how to use and apply fast fMRI to study the brain, we need to think very carefully about the hemodynamic responses and the timescales of the signals that we're actually measuring. Similarly, even if you're studying something slow, it's really nice to just acquire more data. But the example I just showed you actually wasn't completely fair because there are some trade-offs as you go faster. And in particular, the extra data points that you acquire aren't actually statistically independent. And so even if you acquire twice as much data, you don't actually have twice as much information available to you in your analysis. So what I'm planning to go over today, it's relatively brief, but I'll focus first on just, things that have changed in acquisition for fast fMRI. I'll talk about the hemodynamic response, since that's the basis of the signal that we're measuring, and we'd like to understand the temporal properties of those signals. I'll spend a fair amount of time on understanding how noise and signal change with sampling rates. And then I'll talk briefly about current applications of fast fMRI. And one interesting thing about this talk, I think, compared to others in the session, is this is maybe one of the newest developments in fMRI, and as such, some of the aspects of this work are less mature, and there's a lot of really exciting and active development in this area. So I'm going to try and also call out some abstracts that are here at ISMRM, sort of showcasing a lot of the ongoing work that's happening that you could go and look at for more information about kind of how these methods are developing now. And then I'll end with just some limitations, confounds to keep in mind if you do this type of imaging, and a bit of discussion of what may be possible in the future as we push to faster temporal resolution. So I'll start really briefly on the acquisition section, and we actually heard a fair amount about this in previous talks, so I won't spend too much detail. But I think it's worth remembering that it's actually been possible for a long time to do high temporal resolution imaging. If instead of taking the time to image your multiple slices, if you just acquired one, you can actually go really fast, maybe in 80 or 100 milliseconds. And so this has been used since the 90s, actually, to try and capture faster timescale dynamics. But unfortunately, for most applications, you probably want more than one or two slices. You actually want, if possible, whole brain imaging. And so the real kind of changes in acquisition that have been very useful for fast fMRI have been ones that allow you to do fast imaging with this whole brain coverage. So one of the first developments that I'll just mention briefly is these related imaging approaches, MR encephalography or inverse imaging, which are sort of an extreme case of parallel imaging that essentially, would use coils as detectors. They're kind of inspired by MEG or EEG. And as such, they achieve very high temporal resolution. So you could image, for example, with a TR of maybe 100 milliseconds, but this comes with a real cost of spatial resolution. And so what's become now very widespread since this development of simultaneous multi-slice imaging is the use of SMS to acquire multiple slices simultaneously and thus achieve very fast imaging. So instead of taking the time to step through your slices one at a time, if you now were to acquire, for example, in this example, these two orange slices simultaneously, you end up with an image where the slices are overlaid onto each other, but it's actually possible to go back, untangle these two images, and now if you're acquiring two slices at a time, you image twice as fast. And you can keep accelerating in terms of acquiring more and more slices at a time. And through that, it's turned what used to be, for example, a 2-second scan into a 300 millisecond scan. So there's a few good reviews on these topics that I would suggest to look into more if you want to kind of understand the development of these acquisition methods. I'll just talk really briefly. If you were interested in doing high tempo resolution imaging, what might you need to think about as you're setting up your scan? So if you start, for example, in this cartoon with just 10 slices, the first thing you would decide is what should your multi-band factor be. And so if you went, for example, to a factor of 2, now instead of acquiring one slice at a time, you acquire two slices at a time. And so you might get these two blue slices, and then these two green slices, and then the two pink slices. And so in doing that, you've accelerated by a factor of 2. You could also imagine going to a factor of 5. In practice, that would work well. In this example cartoon, this would work very badly. And the reason is that in this case, the slices are very close to each other. And so ideally, you want a multiband factor that lets you accelerate to a reasonable level, but without pushing things too far, such that the information that you're trying to untangle from these different slices is too correlated and close in space. So in practice, we go up to about SMS 8 if we need to push the tempo resolution. Certainly, if you do less, if you can afford less, you'll also perhaps be able to reduce your noise as well. The other factor to set when you're setting up a fast scan is the shift. And so this shift you can see here is the field of view over 2. So this second slice has been shifted by half relative to the first slice. Some sequences will set this for you automatically. If they don't, it's worth spending the time to just try out a few different ones, look at your images, and get a sense of what's actually working well in practice, because I haven't found that there's sort of a reliable general rule for this. A third factor that I bump into a lot, actually, as I really try and push the tempo resolution is that you actually bump up against the SAR limits for what you can achieve in the scanner. So there's a couple of ways to deal with this. You could either adjust your verse factor. This will let you reduce SAR with the cost of some distortion. Or you can reduce your flip angle. But it's often the case when you're imaging fast that you're already thermal noise dominated, and there is, again, an increase in noise as you reduce your flip angle. And then finally, evaluating image quality is very important. It's a good idea to look at these data. There's often options to select different kernel sizes for your reconstruction in SMS, and the larger ones will work better. They come at a cost of longer reconstruction times. So sometimes I'll finish a scan at midnight, and the images will actually reconstruct overnight and become available at 7 in the morning, just squeaking in before the next scan starts. So that's something to be aware of. Another particularly challenging thing for SMS is it's actually really hard to evaluate the performance of your scan in phantom data. These methods don't always perform the same way in a sort of spatially homogeneous setting. And so it's nice to actually do a lot of this testing on a human brain where you get a kind of more accurate sense of what the images would look like in practice. Okay, so whirlwind overview of what you might do if you wanted to acquire fast data. So then the bigger question, I would argue, is, well, how fast are the actual signals that we're measuring with fast fMRI? So as the great talks before have already kind of shown, what we measure in bold fMRI is not the neural activity, it's the hemodynamic response to the neural activity, which we detect as local blood oxygenation changes. And the canonical hemodynamic response function, this is the one that's often the default in any software package that you might be using, is quite slow. So slow not just in the sense of time to peak, but in that it has this broad shape, which is blurring away a lot of the fast information. So why would this matter? If you imagine that you have very slow neural activity oscillating at 0.05 hertz, so a period of 20 seconds, and you convolve with this slow response, that's fine. You get a very nice large fMRI signal out. If you convolve now still quite slow neural activity, 0.2 hertz, so oscillating at 5 seconds, there's now this really significant attenuation of the fMRI signal because of your slow hemodynamic response. If you speed up now just to 0.5 hertz, in the neuroscience world, this is still a very slow signal on a period of 2 seconds. At this point, based on what we predict from these canonical models, you wouldn't expect to see an fMRI signal at all. And so in part because of this, there was a bit of a lag towards taking up these fast methods and actually using them to look for fast signals, due to this presumption that the fMRI signal itself is quite slow. But what was intriguing is there started to be resting state bold fMRI studies, which would look across different frequency bands, and actually they started to find a lot of interesting structure present above this typical 0.1 or 0.2 hertz signals. So this was the sort of first initial suggestion that there might actually be fast dynamics present in fast fMRI data. One challenge in resting state, of course, is that we don't actually know what the neural activity is, so it's difficult to establish the direct link between the neural signals and the bold fMRI signals. And so we turned to task imaging studies where we would, for example, use a visual stimulus to cause a neural oscillation that we know the frequency of. And then we could measure, for example, at 0.2 hertz or 0.75 hertz and find that, in fact, it's actually possible to detect surprisingly fast oscillations of up to 0.75 hertz directly in the fMRI signals. And so this is kind of interesting. It's exciting that we can detect fast signals, but it's also sort of surprising based on how we typically model and understand the hemodynamic response. And so there's really kind of two, I think, major factors that are contributing to these really fast signals that we're measuring. And one is that it's actually very well established that the hemodynamic response function is not linear. And what I mean by that is that the shape of this function really depends on what the brain activity is doing. And this shape will vary as you vary your stimulus or vary your experimental design. And so an example of this is just as your stimulus gets shorter and shorter, you can see here, if you show a visual stimulus for 4 seconds versus 167 milliseconds, your response actually gets faster and faster. And so the original studies, which did a great job of mapping what these hemodynamic response functions look like, they also used very slow stimuli. So for example, an 8-second stimulus. In most contexts, however, particularly I think in the resting state, neural activity varies very rapidly. It doesn't just go on for 8 seconds and off for 8 seconds. And so these fast, nonlinear hemodynamic responses may actually be a good representation for what's happening in typical brain function. The second important point is that we also know that the hemodynamic response isn't the same across the brain. It varies a lot over space. And so because of this, you can imagine that if you were imaging with large voxels like this, you might see this large, slow hemodynamic response. But if you image with small voxels with variable hemodynamic response functions, you might pick out really fast ones and really slow ones. And so because of that, high spatial resolution imaging, like we saw in the last talk, can actually buy you better temporal sensitivity. There's an interesting poster by Jingwan Chen on Wednesday that talks about this relationship of the high spatial resolution imaging to fast hemodynamic responses. The other sort of intriguing aspect of fast hemodynamic responses is that they may actually buy you better specificity. So this was also mentioned briefly in the last talk, but essentially it's been shown pretty reliably now that the deep gray matter has really quite fast bold responses, whereas many of the large slow signals that we were seeing were actually coming from vessels at the surface of the brain. And so as you move to higher spatial resolution, you could become more specific to those gray matter voxels and detect faster signals. So to summarize this part, HRFs are actually often faster than previously thought. What's nice is that this is particularly specific to the gray matter. And so this may be a sort of broadly valid model to study many different kinds of neural activity. All right. So a second really important aspect is how noise and signal change with sampling rate. And so there's many sources of noise in fMRI, but two particularly important ones are thermal or white noise, which can relatively increase in fast imaging, and then physiological structured noise. And this doesn't necessarily increase, but it really changes qualitatively when we image quickly. And so you can imagine if you had a fast oscillation, maybe from your heart rate, and you were measuring it slowly, this fast oscillation would alias down into your signal. But if we image quickly, we actually now can capture these physiological fast oscillations directly in our data. And by doing so, we change a lot about how our signal and noise are represented in the data. So this, for example, is data that we acquired with a TR of 227 milliseconds, and you can see these really distinct peaks for the respiratory signal, its harmonic, and the cardiac signal, whereas the cardiac harmonic is actually still aliasing back down into our spectrum. And so it's important to be aware of the fact that now that we have this really broadband signal, the techniques that were developed for conventional fMRI, which have been really well validated and developed for those data, rely on assumptions that aren't quite as true in the case of fast fMRI. And so a very good example of this is the fact that nuisance regression actually can introduce errors when you're working with rapidly sampled data. So if you have a gray matter signal in the simulation and a nuisance regressor that has the same low frequency noise, but phase shifted high frequency noise, regressing that out can actually cause you to increase high frequency noise into your gray matter. And it can cause, very importantly, if you take a simulation, you can see that if there was no functional connectivity present in high frequencies, doing this type of nuisance regression can actually introduce fictitious correlations. And so it's very important to start to become aware of how regression techniques developed for slow data might change or alter the properties of your data when you work with faster signals. And there's also a poster on Wednesday that takes new model-based physiological noise removal approaches to selectively remove physiological noise in these frequency bands while preserving the background spectrum. The other thing to really be aware of is autocorrelation as you move to faster imaging. And this is this idea that even if you acquire more data points, this data point, you actually probably could have predicted it from the one that came before. And so these points aren't independent in time. And this becomes really increasingly apparent as you move to faster sampling. So Bowman et al. Showed, for example, that when you move to lower TRs, you need higher order autoregressive models to represent the variance in your data. And you can see this also if you look at the autocorrelation functions across different TRs, that there's essentially a broadening of this that you need to be careful of in fast fMRI data. And so this is important because these noise statistics are part of the GLMs that are conventionally used to analyze your data. And so whether there's autocorrelation present or not will actually influence the amount of certainty that you should have about your conclusions. It's also worth looking at your data and just being aware of the kinds of image artifacts that can appear. So for example, there can be slice group artifacts. You could have leakage of different slices that were acquired at the same time. Even if you don't have leakage, 5 slices acquired at one moment in time will contain the physiological noise from that moment in time. And the ones that are following will have physiological noise from a different moment in time. So there's also true signals present in these that are different depending on which slice group they were acquired in. And then also as you move faster and faster, you start to not just be sensitive to bold, but also to things like inflow. So when fresh blood flows into your imaging volume and it hasn't reached steady state, you're going to become increasingly sensitive to signals, particularly in the edge slices of your volume that aren't related to bold and really reflect other dynamics taking place. So on the analysis side, it's just important to keep in mind that the statistical properties of fast fMRI data are really qualitatively different. There's a lot of new information that's present, but the noise structure is different, and it's worth keeping that in mind as you analyze your data. There can be a relative increase in noise, but you have so much more data that usually it more than compensates for that as long as you take it into account in the analysis. And the physiological noise is different, and so kind of standard regression approaches may not be appropriate. And I recommend this review by Chen et al., which is a really useful kind of overview, including simulations of how standard techniques may or may not apply in the case of faster data. Okay, so applications. I wanted to just briefly highlight what fast fMRI could potentially be used for because it's still relatively new and I think there's a lot of space to be explored with these methods. One particularly interesting study showed that when you sample quickly, you can actually detect activity onset times with really high precision, so on the order of, for example, 100 milliseconds. It's important to be aware that because hemodynamic response shapes are different in different brain areas, you need to also control for local waveform differences. And so this study did this by kind of separating different hemispheres and showing that they could detect different onset times. There's also a lot of interest in dynamic analyses of resting state fMRI data. This great benefit of sampling really quickly is that now, for example, maybe a 30-second window is actually sufficient for you to start to estimate local correlation patterns because you have so much more data now present in that 30-second window. So that can potentially allow dynamic resting state fMRI to be performed at faster timescales. We've been working on fast fMRI in the context of sleep. So you can see as people transition between different vigilance states, there's actually clear differences in the spectral content of the resting fMRI signal with a strong increase in 0 to 1 hertz power in the sleep state. And also, even if you're not studying fast activity, perhaps you just want to understand neurovascular coupling and physiology, imaging at fast rates can let you really study these responses with high precision and high temporal information. This is just kind of a selection of what's possible, but I think actually the really exciting applications of fast fMRI are also in progress right now. And so there's quite a few of these at ISMRM that I'm interested to check out. There's temporal dynamics of attention, connectivity fingerprints with really fast sampling, audiovisual processing. There's simultaneous EEG and fMRI of alpha and the thalamus, and we'll also be presenting some fast fMRI work during sleep. So just to wrap up, it's important to keep in mind the ultimate limitations of these methods. And so ultimately, we are measuring the bold response, and it's very exciting that we've gotten up to 0.8 hertz. Maybe we can go to 1 hertz, but fundamentally, we are measuring the vasculature. And so it's probably unlikely that that's oscillating at 10 hertz or 20 hertz. There's other interesting, abstracts here talking about maybe elastography or other approaches you might take to measure quickly. But with, current bold-based SMS, we're going to hit a limit at a relatively soon point. It's also important to keep in mind physiological noise. So even as we try and increase our signal strength, physiological noise will also scale with signal. And so we're going to bump into ultimate limits in terms of the really large contributions from respiration and cardiac to the data that we acquire. And it's also really important to keep in mind that these signals actually are very small. So even, for example, though we found that we could detect 0.75 hertz oscillations, this y-axis is 100 times smaller than the one over here. And so there's still a large attenuation factor as you go to faster and faster timescales. This took a lot of averaging, and you either need a lot of data to work with, or you need really careful noise removal approaches to start to look in these high frequency bands. And it's also really worth keeping in mind as you do this that firstly, this is still a pretty new approach compared to a lot of fMRI. And so things are changing quickly. It's worth really sort of reading and being aware of new technique development and new problems that people are discovering. It's also really important to account now, not just in the sense of kind of a time series, but really thinking about the frequency structure of your data and the frequency properties of your noise. Consider the role of physiology in your study. It's often the case that physiology is correlated to the task or that it dynamically varies over the resting state, and it's a huge contributor to the fast signals that we see in fast fMRI. And finally, it's worth taking extreme caution with GLM-based statistics. So they're very appealing because they've been very well developed, but the software packages that are out there are really optimized for low temporal resolution. And so until models have been developed that really kind of bring us back to a white noise regime, it might be appropriate to start with non-parametric testing. In that case, to try and minimize the assumptions that you're making about the statistical properties of your data to try and ensure that the patterns that you see are truly there. So I'm very excited about the future possibilities for high tempo resolution fMRI. Fundamentally, what we want to study is fast, and so it's great to image quickly and actually sample it. But also, even for people studying slower things, it seems that HRF itself is actually often fast, particularly in resting or naturalistic contexts. And it's also very appealing that gray matter has the fastest responses, so you can potentially buy spatial specificity by sampling faster. And so just to conclude, basically, even though in the past we've mostly been focusing on low frequency bands to look at neural activity, there's really rich dynamics present in fMRI signals, and we can kind of expand that frequency spectrum out at least to 0.75 hertz. And I'd encourage you guys, I want to end on this slide. There's lots of interesting abstracts here, and I would encourage everyone to go check them out and see what's happening in acquisition physiology and applications of fast fMRI. Thanks.